Hello, my name is Jason Siddiqui, and today we're going to be discussing the topic of propagule pressure. A propagule is simply just a set of individuals released into a non-native environment. Propagule pressure is the quantity, quality, and frequency of these propagules or non-native species introduced into an environment, and propagule pressure is sometimes referred to as introduction effort, but for this presentation we won't be using that term. Propagule size is considered the number of individuals released in one event, and propagule number will be referred to as the number of propagule release events. Propagule number is also sometimes referred to as introduction frequency, but again we won't be using this term for simplicity. And here you can just see in these images we have two images of zebra mussels, which are propagules that successfully establish themselves in the Americas, but we'll speak about these later. We're first going to discuss the factors that affect propagule pressure, then we will discuss the impacts of propagule pressure and how we can use this information to our advantage in modern invasive species management. Calculating propagule pressure can be somewhat of a mixed area with many different complex equations being used to give it a quantitative value, some of which account for different factors such as propagule quality, as well as specific biotic and abiotic factors, while others do not. From a blanket perspective, however, propagule pressure can simply be referred to as a function of the number of propagule introduction events with the size of these propagules, with these terms being propagule size and propagule number as mentioned previously. When studying propagules, we can refer to the origin of these propagules as the donor region, as well as the location of where these propagules are received as the recipient region. This image in figure 3 from Alex Pichot's analysis of propagule pressure does a great job of explaining these terms. It shows how these differing components of propagule pressure impact establishment frequency, where both the propagule number and propagule size have an impact on establishment frequency. As you can see on the left, you have a large propagule size and propagule number. In the middle, you have a large propagule number but low propagule size, and on the right, you have a low number but large size. You can see that these work in conjunction together to create a large establishment frequency. The region and transport factor are super important when it comes to studying propagule pressure. When simply referencing the donor and recipient region, similarity between these environments can increase the effectiveness and chance of establishment under propagule pressure. If the recipient and donor region are dramatically different, let's say for example one goes from a humid and hot environment with fresh water to a dry and cold environment with salt water, establishment may never occur no matter how high the propagule pressure is on the recipient region. Another small note is the propagule's activity in the donor region. If the propagule is in high density in the donor region, it will likely come in larger propagule sizes, increasing pressure. Now we'll talk about transport vectors. The transport vector is extremely important in propagule pressure success. When in transport, propagules are put under stress where they will likely face high mortality or be released in an unfit shape to successfully establish. This can be mitigated if the propagules are high in density and genetic diversity, or if the transport vector is simply more suitable for survival, increasing the probability of the propagules reaching the destination and surviving the transport. The graph here from Daniel Vieter's research on the introduction of different types of plankton via ballast water on ships showcases this perfectly, showing how the type of vector can dramatically impact the propagule survival during transport, with the cargo hold preserving density and diversity a lot better than the deck tanks. It also shows how starting with a higher initial population density and diversity increases the likelihood of the propagule reaching the recipient region. Lastly, and arguably most important, is obviously the recipient region. The recipient environment is obviously likely to resist non-native species establishment, which is why disturbance in the recipient region can foster propagule establishment to such a high degree. Disturbance in a recipient environment can decrease the biotic and abiotic resistance put on the non-native species, allowing them more access to resources and increasing their likelihood of establishing. Clark and Johnson found evidence of this with the invasive propagule red rust bryozoan. Bryozoan was unable to normally establish under just high propagule pressure, but had success when the high propagule pressure was in conjunction with a disturbance to native communities. Extremely high propagule pressure can occasionally overcome a lack of disturbance, however. For example, Warren and her colleagues found that simply a drastically large propagule pressure of Japanese stiltgrass was able to outcompete and establish itself in native deciduous forests despite no disturbance in the forest. Overall, we can assume that disturbance might be essential for establishment under low propagule pressures, but has a weaker overall effect when the propagule pressure is very high. So we mentioned how propagule pressure is a function of quality, quantity, and frequency of propagule introduction. So now we're going to talk about one of the most overlooked aspects of propagule pressure, propagule quality. Propagule quality can be referred to as a function of the physical condition, morphology, as well as behavior of these propagules. In some trials, propagule quality is shown to have a larger impact on establishment than other factors, such as the level of disturbance, propagule size, or propagule number. Favorable phenotypic traits have also shown to increase establishment chance, as shown in an analysis by Burgess and Marshall in 2011. Burgess and Marshall studied the phenotypic effects of the marine bryozoan compared to other factors we've discussed, and found that having the trait for a short egg dispersal time had the largest influence on establishment and was necessary. For example, increasing propagule size and number on a population that all had long dispersal times did not increase establishment because they did not have the phenotypic trait of short dispersal times, which appeared to be necessary for possible establishment. Propagule behavior also plays a large role, primarily when referring to animals. Propagules that exhibit cooperation tend to lead to increased density, which increases their mating ability to effectively garner for resources, as well as ability to survive predation. This would allow propagules to overcome what we call Ali effects, which we will speak on later. The illustration here shows an experiment testing the effects of propagule quality, disturbance, intensity, as well as propagule pressure on C. cylindricea, a type of algae. The result of this experiment suggested propagule quality played a larger role over disturbance and size and establishment probability of this algae. It's important to note that propagule quality is not technically an aspect of propagule pressure using our calculation of propagule size and propagule number, but it is extremely important and almost always mentioned in research. So we've discussed cofactors that alter the effects of propagule pressure, such as propagule environments, disturbance vectors, and quality. Now we're going to speak on propagule size and number, the main aspect of propagule pressure. As you'd expect, larger propagule size and number, meaning larger propagule pressure, results in increased probability of establishment. But you may wonder why all those other cofactors having an impact, with all those other cofactors having an impact, why this is still so important. When propagules are introduced into an environment, they're likely to experience what we call stochastic shocks, which are unpredicted events that alter the environment and population size. These could be disturbances or are disturbances in themselves sometimes. Larger population sizes are more likely to be able to withstand these stochastic shocks. For example, if an event that wipes out 90% of the propagule population, the likelihood of the species survival is larger if the released propagule size is large. Larger propagule number also allows for larger ability for the propagule to be placed in a suitable habitat for establishment. A location differential of just 100 meters could be the decision maker in establishment or not for some of these non-native species. This graph here from Mark Ura shows just how increased size impacts the probability of establishment. It also shows 3 different environments, showing how finding a suitable environment also plays an impact on establishment, which here would be propagule number, showing how a large propagule number you get to multiple different locations, finding a suitable habitat. Lastly, like we said, a large propagule size is suitable to overcome Lee effects. Now you might be wondering, what are Lee effects now that I've mentioned them twice? Lee effects are the reduction in per capita growth rate of a species at low population density. At low population density, a propagule might struggle to do things like find partners to reproduce, effectively forage for nutrients, and even form suitable mutualistic relationships needed for survival. This plays right into our discussion on behavior and location as well, as we discuss the importance of a large population density of the propagule. A real-life example of where a propagule can't form necessary mutualistic relationships could be with plants that require pollinators. A single plant may not be attractive for a pollinator, but multiple plants in a small area showing high density is much more likely to attract pollinators, a necessary mutualistic relationship. This image here isn't from an experiment, but just shows a general illustration of how Lee effects works, where the growth rate is contingent on the population density, and at low densities, the growth rate tends to be extremely low and sometimes even negative. Now let's discuss some successful propagule studies and learn which aspects of propagule pressure impacted them. Firstly, we'll discuss the zebra mussel, which was shown previously in this presentation. Zebra mussels were introduced into the Americas from Eurasia through ballast water ships that were dumped into the Great Lakes in the 1980s. Marjorie Warnham and her colleagues that researched the zebra mussel invasion found that although the size of each propagule was likely low as these ballast waters were sent through multiple series of filters prior to being dumped, multiple introductions is likely what allowed for their establishment. This would be a case of a high propagule number and a low propagule size, allowing for potential establishment and the main source of the propagule pressure. Now, on the complete contrary, is the Tretescantia leaf beetle. The Tretescantia leaf beetle displays a complete opposite form of propagule pressure that was successful than the zebra mussels. These beetles were introduced in New Zealand as a biological control agent to suppress the spread of Tretescantia, an invasive wildflower. The leaf beetle was introduced in large sizes and a relatively low propagule number, with them only being introduced in a couple locations in the country, which allowed them to overcome Lee effects and successfully establish. It can be assumed that the New Zealand researchers chose a larger propagule size as these beetles reproduce sexually, so a high propagule size would make them more suitable for overcoming Lee effects and finding suitable partners. Before we talk about understanding propagule pressure to manage invasive species, we must first understand how these propagules establish at certain propagule pressures. This is a dose-response curve, and here we see that propagule establishment probability either grows at a linear rate or exponential rate with pressure increase. At exponential growth, there appears to be a threshold at which decreasing the propagule pressure has no impact on changing the establishment frequency, while if the species responds linearly, any biological control will have a proportional decrease on the establishment success. Propagule pressure has been used to manage invasive species many times successfully in the past, and prevent new invasive species as well, with one of them being the Tretescantia leaf beetle, as we discussed prior. When primarily referring to biological control, we have to look at propagule pressure as well as run necessary environmental risk assessments to judge the likelihood of the biological control agent establishing, as well as if it will stabilize the ecosystem or potentially even make it more erratic, acting as an invasive species itself. Another example of the use of propagule pressure in successful invasive species management is with the control of the cotton cushiony scale as seen in this image, which was a type of invasive scale that was outcompeting native species and ruining agriculture in California. Researchers were successfully able to introduce both the Vidalia beetle and parasitic fly, which were both direct predators of the cotton cushiony scale, and they successfully stopped the population growth. Now let's talk about the use of propagule pressure against other invasion theories, as well as the flaws that come with propagule pressure. Another unrelated invasion theory to propagule pressure is the natural enemy hypothesis, which is the theory that invasive species are less affected by native predators than are native species, which promotes their establishment. When we compare this to propagule pressure, we see that the enemy hypothesis focuses on species behavior and ecosystem interaction within the environment. This is very different than propagule pressure and points out one of the potential caveats of only looking at propagule pressure, as propagule pressure is essentially just a numbers game and gives you a perspective independent of alternate factors. A big problem with propagule pressure is also the difficulty in tracking propagule pressure. During an active invasion, tracking propagule pressure has shown to be extremely difficult, and getting data on propagule pressure retroactively is also nearly impossible. Many researchers such as Collodi and Lockwood have suggested using propagule pressure essentially as a null model when studying species invasions, as it provides a great general view that can be built on with further analysis. The image of the dose-response curve here is put twice intentionally, just so we can focus again on the flaws of propagule pressure. While these graphs are theoretical, in practice, these results are very unlikely to be true, as propagule pressure fails to account for things such as biotic and abiotic factors, disturbance, predation, and other factors in the environment, as we discussed, showing that this graph is likely to be unrealistic in a true environment. And just to sum everything up, despite its flaws, propagule pressure is a very good tool in measuring how vulnerable an environment will be to invasion, as well as how we can prevent invasive species establishment in the future. Knowledge of propagule pressure can be and has been used to accurately explain species invasions on multiple different occasions, as well as how propagule pressure can be used in effective biological control acts. And lastly, here I added a picture of the invasive hippos in Colombia, which were previously imported over by wealthy organized crime leader Pablo Escobar and have since grown in population drastically because they have no natural predators in the area, and the moist climate has allowed them to survive and reproduce with nearby food supply. Thank you so much, and I hope you have a good day.